Hi, everyone. Welcome to the In Silico Drug Discovery Workshop. We're very excited to have you all join us today. I'm Serene Markosian. I lead the Assay Guidance Manual program here at the National Center for Advancing Translational Sciences, part of the National Institutes of Health. So we have a super exciting program for you all today, but would like to start with our introductory comments. And so for that, I'd love to introduce Dr. Joni Rutter, who is the director of the National Center for Advancing Translational Sciences. Joni, we're super excited to have you here to open up this workshop. So the floor is yours.
Okay, thank you so much, Serene. In addition to being our NCATS editor-in-chief of the Assay Guidance Manual, Serene is just an amazing ambassador for translational science. So thank you, Serene, for the introduction. Also, big thanks to our incredible organizing team from NCATS, especially Abigail and Hannah, and also our BARDA colleagues. And I'm glad Kimberly will follow my remarks too. I also want to give a shout out to UC San Diego and UNC Chapel Hill. All of your efforts in supporting this meeting and bringing this together for what promises to be a pivotal meeting, it's super exciting. And thank you all for joining us today. This is the October AGM in Silico Drug Discovery Workshop, and it'll be going today and tomorrow. And if you don't know about AGM in general, this program, it's really a world-class resource to look at best practices in translational science, and it provides robust guidelines for preclinical research, assay development, data analysis, and so on. And in this way, when we have discussions very deeply in these areas of science, we ensure that the findings are reproducible and they can move smoothly from bench to bedside. So these workshops really play a key role in ensuring that we have those critical discussions within our scientific community to make that happen. And this workshop will focus on preclinical innovation, which is one of course, the critical steps in the translational pipeline. And it'll have a special emphasis on how data science and AI and ML methods are really revolutionizing drug discovery. And at NCATS, we believe that team science, having these meetings together with biologists, chemists, data scientists, informaticians, statisticians, all working side by side to push the boundaries of what's possible. And so we think it's really critical to have these meetings to bring all of those disciplines together to really look at how we can divine those best practices. And as we look to the future, of course, AI/ML is really pivotal to accelerating drug discovery and improving translational science. And our teams at NCATS are applying AI/ML to enhance things like high throughput screening by detecting assay artifacts and optimizing compound properties like metabolic stability or absorption or toxicity, for example. And these methods also predict synergistic drug combinations for diseases like cancer, rare, and infectious diseases. And they're also advancing the next generation modalities such as molecular glues or proteolysis targeting chimeras or PROTACs, targeted antibodies or nanobodies. So these are all critical modalities that are really coming upon the scene, and how we study them, how we generate them, and how we think about using them to translate them into clinical practice is where this is all starting. And so for example, one of the ways NCATS applies AI/ML is in improving high throughput screening outcomes by detecting potential assay artifacts. And this helps ensure that the data we rely on is robust and accurate. And that's, as you know, very critical for advancing the promising candidates forward. We have a really high failure rate when we move compounds through the translational pipeline. And this is one of those areas that we hope to really address so that we can bring down that high failure rate. We're also thinking about applications in quantum sciences and have a few challenge competitions out on the street on that. I'm happy to put those in the links when I'm finished here. But the 3 areas of interest for the current quantum challenges include quantum algorithms for drug discovery, quantum algorithms for clinical risk predictions, diagnosis and therapeutics, as well as quantum algorithms for biomedical imaging and genomic analyses. But one of the key focuses of this meeting is to identify and address gaps in the translation of in silico models into effective therapies. And just as we've seen challenges in translating data from biochemical assays to cell-based assays, from cells to tissues, from animals to humans, similar gaps exist in the progression from in silico models to then the real-world applications that lead to effective treatments. And each stage of the drug discovery process requires, really those specific types of models, and the predictions made by these models has to align with the biological complexities at that stage. And so as we move from computational predictions to experimental validation, we need to carefully evaluate which models and predictions are best suited for each step in that pipeline. And then we also need to consider what new models and approaches are required to improve translation and ensure that those in silico insights lead to those tangible therapeutic advancements. And so that's really what I'm super excited about for this meeting is really getting some good insight from the people we have at the table to talk about this. And if that wasn't exciting enough, I want to take a moment to acknowledge something truly extraordinary. It's not every day that we get to hear from a newly minted Nobel laureate. And so we're really grateful to have David Baker join us for this meeting. And he'll talk tomorrow about his work. As you all know, David Baker was awarded the 2024 Nobel Prize in Chemistry for his groundbreaking work in computational protein design, along with Demise Hassabis and John Jumper for their protein structure prediction work too. And, this field is fundamentally reshaping our understanding of biology, and it's opening new doors for therapeutic development. And so I really look forward to these talks as we start to hear more and apply those learnings to what we're going to do in the coming years. And we also have brilliant experts from across the translational science spectrum who are also going to cover essential topics from AI-driven drug discovery to the preclinical and practical challenges of translating in silico models into effective therapies. And again, this is important to note because the Nobel Prize in Physics was awarded to John Hopfield and Jeffrey Hinton for their foundational discoveries and inventions that enable machine learning with artificial neural networks. And so while it's really fun and fantastic to celebrate those achievements, it's also equally important that we build on them, identifying gaps in the translational process and finding ways to address them. So how do we ensure that what works in the lab works in real-world settings? How can we use AI to improve the reproducibility and reliability of drug development? These are some of the key questions that I look forward to hearing about over the next 2 days for this conference. And this workshop really represents the collective efforts of institutions across the public and private sectors that are exemplifying the power of collaboration and team science that can really help us drive the science forward. So as we kick off the meeting today, the only thing I ask of you to keep thinking about in the back of your mind that moves to the front of your mind is please don't lose sight of the real goal here. The work we do here today will directly shape how quickly and effectively new treatments can reach patients and ultimately save those lives. And we like to think that our big goal is to bring more treatments for all people more quickly. So thanks very much for joining today and tomorrow, and let's get started. Kimberly, I'll turn it over to you, or Serene, if you'd like to introduce Kimberly. Thank you.
Yes, thank you so much, Joni, for that exciting opening remarks. So the next person for the opening remarks is Dr. Kimberly Sciarretta. Kimberly is the acting director of the Division of Research and Ventures, DRIVE, which is part of the Biomedical Advanced Research and Development Authority, BARDA. And she's the branch chief of DRIVE's launch office. Kim, thanks for joining us. The floor is yours.
Thank you, Serene. Unfortunately, it doesn't look like I can go on video right now. I'm not sure if that can be fixed, but I will present my slides, and I hope my laryngitis that I'm recovering from doesn't interfere too much with this presentation, but the slides can help counter for that. So I'm excited to be in this position and kicking off this in silico drug discovery meeting with our colleagues at NCATS. I've heard that we have quite a response to this meeting, and this is truly a reflection of how broadly resonating this topic is and impactful this topic is to all of you. But I would be remiss if we didn't thank and acknowledge all of the workshop organizing committee and all the efforts put into planning this meeting. And that's with our colleagues at NCATS, BARDA, and then UCSD and UNC. So before I discuss some of our interests in this space and why we're excited about this meeting, I wanted to provide some background on BARDA in case you were not familiar with us. So we're the Biomedical Advanced Research and Development Authority. We sit within the ASPR, which is the Administration for Strategic Preparedness and Response, which is within Department of Health and Human Services, HHS. I know, quite a mouthful. ASPR's mission is really to prepare and respond to public health emergencies, and a big part of our work was responding to COVID-19, but we also respond to other public health disasters like hurricanes and forest fires. So BARDA's role in this is to develop the medical countermeasures and make them available for these public health emergencies. And what do I mean by medical countermeasures? Because that may be more of a U.S. government term. We mean diagnostics, devices, vaccines, prophylactics, any type of therapeutics and drugs, which is the topic of this meeting. And we're developing these medical countermeasures to respond to any chem bio-red nuke. So you'll see the acronym CBRN, flu, and emerging infectious diseases. And we do this through forming these unique public-private partnerships to really drive innovation. And this has led to the success. Our BARDA-supported partners have had a total of 95 FDA approvals, licensures, and clearances. And these are to develop medical countermeasures that will directly protect Americans facing public health emergencies. But this is a testament to the partnerships of working with organizations like yourselves. So BARDA traditionally focuses on advanced research and development of medical countermeasures. However, we have established the DRIVE division, which plays more in this innovation space, supporting enabling technologies and supporting some earlier research, especially through some of our partnerships with Blue Knight and the BARDA Accelerator Network and our ventures partnership as well. And I will talk a little bit more about DRIVE later in this presentation. But to echo Dr. Rutter in her opening remarks, we are in a unique position here to be kicking off this meeting at a time when just the recent Nobel Prizes were announced in chemistry and physics for just this topic. And although we all understand the potential value that in silico approaches bring to drug discovery, and many of us have personally experienced the lengthy time and resources that go into identification of these candidates, we do see the value that these types of approaches can really bring to identifying safer and more efficacious drugs faster and sooner. And this is critically important for our ability to respond to public health emergencies. But I did just want to show this again and acknowledge again the great work by our colleagues in this space and how impactful this work really is. And we look forward to hearing from Dr. Baker later on in this meeting, as well as all of our other esteemed colleagues here today. But today would not be successful, and tomorrow this whole meeting would not be a success if it wasn't for all of you. Here we are bringing together you as government with academic and industry experts to really explore the future of in silico modeling. And so an ask from you over the next 2 days, we really hope this meeting allows you to foster and form new virtual collaborations as much as possible in this virtual environment. We want you to engage in information sharing. Hopefully this will generate new ideas to address some of the challenges. I hope we can brainstorm these solutions together. Maybe some of these will be the genesis of new program ideas. Hopefully we'll all just gain a better understanding of the current landscape of computational drug discovery. But again, this information will directly help us in the insight that we learn from this in preparing for future public health emergencies. So BARDA currently does not have any open topics or solicit funding opportunities in silico drug discovery. As I mentioned, most of our work is traditionally focused on advanced research and development, and these activities typically occur more upstream with our partners before they engage with BARDA. However, as I noted, We are interested in innovative opportunities and enabling technologies, especially within the Drive division. And there was a notable effort in a program, Redirect, and this was a partnership with Drive and our ChemBio Rad Nuke division, CBRN, that was focused on repurposing drugs to treat conditions as a response to exposure to chemical agents. And there were some efforts in this program related to in silico approaches. But again, there's opportunity, as we've learned from this meeting, to continue to expand in this space. So I did just want to highlight the value in the translational science here of the work that we're going to hear today and how that's going to apply to future of MCM development. And this, again, will directly impact all of you and hope and save Americans when we're facing a public health threat in the future. So one, rapid drug identification is immensely critical when we're facing a public health emergency. And this can be in the design of novel drugs, as well as just identifying repurposed or repositioned candidates to clinically evaluate. The second is we need to increase our ability to apply our learnings from previous threats that we've seen to these new emerging threats. So that touches on our response capabilities, as well as more of a preparedness piece, right? Building out these platforms and these models and these threat agnostic approaches will allow us to be more agile in response to future threats. The other unique thing about developing medical countermeasures for CBRN threats is that we can't always evaluate these products in clinical models, and therefore there's a need for sufficient and equivalent models to evaluate these products even outside of animals. And of course, we're always interested in understanding the pharmacology and toxicology of these drugs. So these are several areas where we see the value of in silico approaches impacting BARDA and impacting the drugs that can get to you all sooner. There are several offices within BARDA focused on medical countermeasure development, CBRN, flu, DDDI, and I will mention Drive. This is the division where I sit. And our mission specifically is to support the development and adoption of these innovative technologies and medical countermeasures. So we don't just support the medical countermeasure development, we also support the enabling technologies that allow these products to be successful. And you can engage directly with us. If you're interested in reaching out at drive@hhs.gov, you can also check out some of our programs at drive.hhs.gov. But you can also engage with the rest of BARDA if you have technologies of interest that you'd like to share with us. Every solicitation topic has points of contacts in it for the topics. Also, you can find points of contact on medicalcountermeasures.gov. We often engage in these market research calls or pre-solicitation calls if related to a topic, but these are typically tailored for the area of interest of that BARDA program. If you have a more general inquiry or technology, I encourage you to check out our TechWatch program. And this is an ability to engage with all of BARDA as well as other U.S. government participants in the meeting, and you can get direct feedback on your technology, and that's at medicalcountermeasures.gov. Oops. So this is how you can connect with us. I do just want to plug that BARDA Industry Day. We've announced the dates for that. It's next summer, June 30th through July 1st, 2025. So I encourage you to follow us on medicalcountermeasures.gov for upcoming information. But thank you again for all of you today and participating in this very important meeting. And here's to a productive meeting, and I look forward to learning from all of you today.
Fantastic. Thank you so much, Kim, for that wonderful opening remarks and letting us know all about BARDA.